So this is a summary of some of the key points. I'm not going over every single question, every different aspects, and common mistakes, unfortunately, that our students make. So just some conventional things. Genus and species names of organisms should always be italicized if you were typing them, and underlined if you're writing them. I'm not a real stickler in this class with that, right? But just so that you guys know the proper way to write an organism name. The word bacillus, although it's a genus name, and again, you would know usually by the context or the fact that you can very easily pick out here that this is italicized, and that's to indicate that it's a name or a genus in this case of an organism. But it is also a common shape. The only two shapes we deal with in this lab, although there's lots of other shapes, right, are the bacillus, the somewhat cylinder shape, and the sphere is what we refer to as cocci. Although there are other shapes out there, right, for us it was one or the other on your reports. Gram staining, gram is usually capitalized. Again, I'm not a huge stickler for that, but if you don't capitalize it, usually it could be, again, confused with a gram, which is a measurement, right, a weight in I forget what system of measurement, right? So for gram staining, remember, it is a really important tool, especially if it's done properly, right? It would help divide up your possibilities in half. And, you know, this is a very widely used technique. And remember, it's based on the cell wall structures of those cells. So it gives us some information. So the gram positives, remember, have the really thick peptidoglycan layer, where gram negatives tend to have a very thin peptidoglycan layer, or if at all. And are characterized by having that outer membrane. These are technique sensitive, right? You have to perform the technique properly, right? So we did positive and negative controls on the same slide. Even then, sometimes different organisms don't stain all that great, and results may be ambiguous. And so that's why it's really important, too, that, remember, we followed up when we did selective media, right, where we were selecting for gram positives on one plate and gram negatives on the other to help confirm those results and give us another test. So you guys performed a whole bunch of staining procedures, right, on your organisms. Gram positives are purple, right, gram negatives are pink. You still need to know this for the final exam, too. You're going to have microscopes you'll need to look at, and you need to be able to interpret results. Know if this is a gram stain, am I seeing gram positive, am I seeing gram negative? Of course, I won't have ambiguous ones there for you, right? They will be pretty clear, good gram stains that you'll have to identify. For negative staining, one of the main reasons we utilize that is usually the one that gives you the best representation of arrangement, but in shape of cells. But any one of your stains that you should have done, right? You should have looked at all of them and taken all of them in consideration in determining arrangement and shape of your cells, right? And you summarize that on your report. So capsules, you were expected to label the cell and the capsule, right? In your drawings and such, you need to indicate too background, right? If you didn't draw a dark background for both negative and capsule staining, you should have definitely included that in your descriptions. Your descriptions should have been something like dark background, clear unstained cells for negative staining. For capsule staining, you should have had, you know, the cell was stained, the background was stained, and the capsule was unstained or a clear halo visible. Right? That's why those had areas for descriptions. Those were the type of descriptions that we were looking for. And again, you didn't have to do your report in color, right? But, you know, you had to indicate colors definitely as it relates to staining. Colors are very important. And then, of course, if you had endospores, you were to label them versus cells. So those of you guys that just had cells, you really should have labeled that cells, right? That drawing, so that we knew it was just cells and not any endospores. I wasn't real nitpicky this time around with that. And acid-fast staining, remember that these tend to clump together, that they tend to be bacilli that are acid-fast positive. Mycobacterium is that genus, right? So if you ended up with a positive acid-fast, if everything else lined up, that was a pretty good indicator of what you were looking at. And then remember, our non-acid-fast are going to be a bluish kind of color, because we use the counterstain methylene blue when we do that. And then the carbofusion makes the acid fast, other than the fact that they clump together and they're rod shaped. They're going to be that fuchsia, purple, pink color, right? And so again, you need to be able to know, again, what endospores look like. And for us, the staining procedure we use uses malachite green, which unfortunately is looking kind of bluish, right? So don't put it in there, girl. Yeah. You need both. So, can you wait a few minutes? You're going to listen to this? Okay, good. Okay. So, yeah, you're going to need to be able to identify and interpret stains just like you did for your unknown on the final exam, right? So make sure you review that information. As well as arrangements, right? Like this is two together, right? So if we show you or ask you a particular arrangement names, remember that's carryover from like the first half of the semester. So organisms that are motile, right? Usually that's by means of flagella. There are others that have other abilities. For ours, that was flagella. We did the motility test. Most of you guys have pretty clear motility tests if it was motile. And you could also look at it under the microscope. But remember, we can't stain it. Why? It would kill it, right? And dead organisms don't move, right? So if you want to observe motility, right, you have to do it under low light conditions to be able to indicate that. And we use a special indicator too, again, so you can really see the growth in those tubes. So again, you'll have to look at tubes and you'll have to figure out, is it positive, is it negative for motility testing, right? All these things you did on your unknown, you're going to do again on the exam, right? You'll have to interpret all tests and understand what's going on, right? So for the thioglycolate media, remember this media is designed to create an anaerobic environment in the bottom of the tube. And this is the one where a lot of people fall short on their unknown reports too. They forget to label the oxygen and non-oxygen zones. It's very important, right? I don't know if I took points off this time, but if you lost points on that page, that might have been a reason why. So you needed to label your oxygen zone, which we know is at the top, right? And your non-oxygen zone, which is towards the bottom. For Delgado, two of these we don't have, right? We don't have microaerophiles, and we don't have obligate anaerobes. It's too hard to maintain the type of environments necessary to grow those organisms. So then you're left with facultative anaerobes, aerotolerant anaerobes, and obligate aerobes. And there was a few of you guys that had obligate aerobes, so because they can only metabolize using oxygen, they stay stuck in this oxygen-only zone until you only saw growth at the very top of your tube. Facultative anaerobes and aerotolerant anaerobes were tough to determine because we didn't look at these the very next day, right? And I even refrigerated, especially our obligate aerobes, so that they would stay at the top. Your facultative anaerobe, if you get a good stab, which actually we had some good examples. I think Untis ended up with a really good one where it looked like this picture, where you definitely saw a lot more growth in the aerobic zone as opposed to the anaerobic zone, because facultative anaerobes can respire aerobically with oxygen and grow quite abundantly. They produce more energy, right? Where they either ferment is the most common, or anaerobic respiration in the oxygen-free zone. Aerotolerant, as the name implies, they tolerate oxygen. Oxygen's not lethal to them like it is for our obligate anaerobes. But they don't utilize it, so you don't see this difference between the oxygen and non-oxygen zone. These guys pretty much just ferment all the time. And we only had one organism in this lab that actually is an aerotolerant anaerobe. And I think Fong had it. You had Anaerococcus faecalis. So this is also... We'll come back to that in a moment. I started another thought, but it's later on in my presentation. Okay, so phenol red broths. The main reason why we do this is to know if they ferment the sugar, and in this case the sugar is glucose, right, in these tubes. But they only slightly are acidic, right? 6.8 will actually turn them yellow and give them an acid reading. So not very acidic. But, you know, enough to indicate there's definitely some fermentation of the sugar happening. That's actually what we refer to as a phenol positive result. Later on, we asked you some questions related to, and as I got to thinking about it, I was like, oh, you know, we probably should have indicated we were talking about acid, as opposed to the other result you could potentially get with your phenol red tubes, which is, remember, it could eat the protein, increase the pH, and you can end up with alkaline results. We didn't use the terminology positive and negative for the phenol red testing, right? We use the coding that you see here. A for acid, G for gas, A for acid, minus for no gas. So although I put answers for you guys on that part of the report in the methyl red testing that ask you the relationship between this test and methyl red as it applies to glucose, I wrote answers for you guys, but I didn't take off points, right? Because I think we really need to clarify what we meant by phenol red positive for that. So, so again, be able to interpret, looking at these tubes, what's happening in these tubes. How do we code it? What does this mean, right? So for the citrate test, I think all you guys got this one in this class. Not so great in my other class. But what we're actually testing for is if they have citrate permease. If they have this membrane-bound protein embedded in their membrane, which allows them to take the sugar citrate from the environment and bring it in. And because in this test, that's the only carbon source, if the organisms don't have citrate permease, what happened to them? They died. They died, right? If they could take in the citrate, then they also need to utilize a nitrogen source in order to grow. And again, there's only one nitrogen source in these, and that's the ammonia. When they utilize that, they change the pH of the media. They actually increase it. And a lot of you guys missed that second question on your report, so you'll notice that I circled it. So they're consuming ammonia, which raises the pH, right? And that's why you get that blue coloration, and that's referred to as positive. So your organism either has or doesn't have citrate permease. This test isn't testing whether they can utilize that nitrogen source. This is a very common nitrogen source. Most organisms can utilize ammonia. The problem is for them, if they can't utilize the citrate, then they're going to die. Right? Doesn't matter whether they could use the ammonia or not, because they've got to have a carbon and a nitrogen source. Make sense? Okay. So our catalase test is again testing for the presence of an enzyme. In this case, catalase. And catalase is an enzyme that breaks down hydrogen peroxide into water and gaseous oxygen. So it does what when you add it? It bubbles, right? Those bubbles, what's in those bubbles is oxygen gas. So organisms that have electron transport chains and are living in an oxygen-rich environment, and especially if they're using oxygen as their final electron acceptor, they will sometimes generate hydrogen peroxide. This is a damaging form of oxygen to cells. So they have to have a way of detoxifying this toxic form of oxygen. So the way they do that is they have catalase, right? They will break it back down into safe gaseous oxygen and safe water. So we have this, right? We're aerobes. We have catalase, right? We can break down hydrogen peroxide. So this is related to our FTM test too, right? Because if your organism is catalase positive, it means it can respire aerobically, right? Or it can live in the presence of oxygen. So again, it makes sense that our one aero-tolerant anaerobe is actually negative for catalase. Doesn't really need this particular protective enzyme. It has the other protective enzymes. And again, it's not using an electron transport chain with oxygen as its final electron acceptor, right? It can tolerate oxygen, but it's not using oxygen. Where your obligate aerobes, right, clearly have catalase, right? And again, a lot of those, right, when you add it, it bubbles like crazy. And then our facultative anaerobes, again, when they're in the presence of oxygen, they're utilizing oxygen, they're using the electron transport chain, they're using oxygen as the final electron acceptor. They're sometimes generating hydrogen peroxide, so they have to have catalase to detoxify. So you guys understand the link between these two? Okay. So then methyl red VP test, so this broth was utilized, this was a glucose-containing broth, was utilized for 2 tests that we did. One of them is the methyl red test, which is a straight-up pH test, but this particular pH indicator, the pH has to be 4.4 or lower in order for it to stay that red color. Otherwise, it turns orange to yellow, which is referred to as negative. This is a very, very high acidic level, right? So very low pH. As compared to your phenol red test, right? Phenol red, if they had acid production, they actually turn yellow. But the pH for that is only 6.8. It's just below neutral in order to turn yellow for phenol red as that indicator. So something that's phenol red positive, right, actually produced acid, enough acid to be yellow in the phenol red test. Is it also going to be methyl red positive? It could be, but it could not be, right? Because, again, 4.4 is a very low acidic level. So in the case of methyl red positive, right, if it has a very low acidity, right, 4.4, very low pH in the methyl red test, chances are it's also going to be positive and produce acid in the phenol red test, because again, that's only a very slight acid production you need to be positive. But the flip side is not also true, right? Because just because it's phenol red acid producer in that test does not mean it's actually producing enough acid, what we call mixed acid fermentation, where you have a lot of acids, to really lower that pH down around 4.4 or lower. Right, because the difference in these tests is that for methyl red, we're looking for mixed acids, for a lot of acid, which really lowers the pH very low, 4.4 or lower. Where with the phenol red test, we're just seeing if they ferment the sugar and produce some acid, right? And so the pH, again, is only slightly acidic there. Does that make sense, Carolyn? Does that make sense to you guys? That was the correlation we were trying to have you guys understand there. And again, you know, that pH of the median, and probably I'll add this in next semester, I'll probably have you guys pH test just the plain up media. In some cases, there is a drop in, again, because we know they eat the glucose if we look at their phenol red test, but then some of them come up negative on methyl red. Well, that's because they're not producing a lot of acid. So they can still drop the pH of that starting media and not create a color change when we add the pH indicator methyl red. And again, the reasoning for that is that's a very low pH. You have to have a lot of acid to drop that low. So we'll sometimes get a decrease in the pH, right? But the important thing is you need an extreme drop in pH. We want to know, did they produce a lot of acid? And usually it's a lot of different acids. And that's why it's called mixed acid fermentation. Not all organisms do that. So with methyl red, with phenol red, you could say, oh, okay, it eats glucose, it produces acid. Okay, let's see how much acid. Let's now do a methyl red test and see if it's positive for that too, right? Does it produce a lot of acid? And there is the potential that it doesn't produce a lot of acid. It's not a mixed acid fermenter. Make sense? Okay. Oh, so one other thing. What are the tests that it's not on the slide. What are the products of fermentation that you can test for with the phenol red broth tubes? Definitely acid, right? What's another one that you guys didn't write? Gas. Right? Don't forget about that derm tube and its ability to detect gas. That is a fermentation product, right? You get acid and gases potentially from fermentation. What's another product that we can test for? Alcohol. And the only test that tests for the production of an alcohol from sugar fermentation, in this case glucose, is the Vogt-Proskauer, the VP test. Right? This is the only one we did that actually detects for alcohol fermentation. And again, for this one, we're not actually detecting the alcohol, we're detecting one of the intermediates, the acetonin. Right? So remember, the reagents actually bind with the acetonin. We had to shake the tube to add in what? Oxygen for this reaction to proceed. Red, again, is the indication. And again, since it's a chemical reaction, we had to wait at least 60 minutes to make sure that we didn't get a positive reaction. Because it was a chemical reaction we're waiting to happen. Unlike the methyl red test, which is just a pH test. That happens within seconds. So our selective media, as I said, it helped confirm, for some cases, gram-positive or gram-negative or different environmental conditions in which organisms can live in. So the important thing with these media, they were all selective and differential. But when you select, right, for a particular organism to grow, say a gram-positive, who are you killing off? Gram-negative. So you stop there, right? If it didn't grow, you can't tell anything else about the organism, right? You can't differentiate any of the differential characteristics if it didn't grow, right? So that's why you had the could not be determined because no growth, right, as one of the choices for your results for the differential component of these plate media we used, right? So one of the selective agents we used were antibiotics, right? It's going to kill off one group and allow the growth of another group. And again, remember that gram-positives versus gram-negatives, they have those differences in their cell walls, and that's why particularly colistin affects the gram-negatives in the way it does because of that outer membrane that they have. And then we also looked at, how resistant they are to high salt concentrations, and for a lot of you guys, I added in the high salt concentration component, right? It's not just that this media has salt. It's the fact that the MSA has a higher than normal concentration of salt. And that's an important factor. Right? So if an organism can't survive, right, as I said, we can't differentiate. If we remove the selective pressure, right, and the plate just contained mannitol, then we could ask the question, does it ferment? Right? But when you have a selective pressure, if it doesn't grow, you can't answer the next question. You can't ask, does it ferment mannitol? So the main reason why we use the mannitol salt plates with that high salt concentrations is we're usually trying to select and grow salt tolerant. The most salt tolerant organisms we deal with are Staphylococci. So our two Staphylococci, Staphylococcus aureus and Staph epi, can be differentiated from each other because Staph aureus, unlike Staph epi, will actually ferment the mannitol. Both of them are salt tolerant, both grow really well on these plates, but that high salt concentration doesn't affect them. But you can distinguish Staph aureus by the fact that it does ferment the mannitol, does produce the acid, so interacts with phenol red, which is the pH indicator in this, and turns those plates yellow. EMB with lactose plates, so this is eosin methylene blue. These are the dyes that are inhibitory to the gram-positives. and help select for the growth of the gram-negatives. They also help interact when the gram-negatives grow. If they ferment the lactose in these plates, it creates color changes to let us know the different degrees of, so for this one it's nice, you get the different colors, right? They'll let you know the different degrees of lactose fermentation. The highest degree of lactose fermentation, usually done by organisms like E. coli, fecal coliforms that we're looking for, is the black to metallic green sheen. Others will have a pink to purple in color. Is somebody stalking me? You need something, Paula? Oh, yeah, no, I'm letting her go today. You don't have anything out right now.
We never get to pick on microbes. I do it so well.
And I noticed it, but I didn't say anything because we're not working with microbes today. Okay.
We're investigating what we do and don't have in the building.
I'm looking for a vacuum line.
Do we have a vacuum line in here? Okay, yeah, I'll smudge that.
You can generate one from a sink, though, you know.
Good.
God, I feel dumb. I didn't know you could do that. Jim just taught me you could do that.
Yeah. It's all about movement.
Okay, maybe you feel even dumber.
Alright, so let's see. Where were we at? Different degrees. So again, remember in the book, it talks about there are different formulations of EMB that some of them actually contain sucrose as well. We buy the one in lab that just contains lactose. We're really only asking that one question as it relates to these plates. And then our last plates are our CNA plates. And CNA, remember, stands for colistin and nelidistic acid. You won't have to spell them, but you definitely need to recognize those antibiotics like you did on your quiz. And those inhibit the gram-negatives. So we're, again, selecting for gram-positives. We're typically looking for, I know I have it on here, staphylococci, streptococci, and enterococci that potentially have hemolytic abilities. Remember, there's two different types of hemolysis. We have beta, which I always remember beta is bad, complete destruction, right, of the red blood cells. So you have complete clearing that you can see here for two organisms. And then alpha is just incomplete, poking little holes. And remember, it looks kind of greenish, the discolorization of the red blood cells in the media. Gamma is no hemolysis at all, right? It will grow, though. So again, that would tell us that this is a gram-positive, but it's not producing any hemolysis, any enzymes that destroy red blood cells. And that's possible. So justification of inconsistent results is very important in your correct identification of your unknown. So I didn't grade you based on whether you actually got the one you were given, like the right answer, I guess you could say. But the fact that what you picked actually matched up with the chart, right? And if you had some inconsistencies, that you justified them, right? And if you read your report, it said that you should have discussed it with me too, if it didn't match up. Now, most of you guys, I checked with you to make sure you had the right one. If you slipped through my cracks, again, you know, as long as everything matched up, you were fine. If you have questions on any aspect, make sure that you come see me. Careful note-taking, right, is a very important scientific tool, right, and in life in general, right? And so every little thing that you could possibly write down. And I think most of you guys and some of you guys are really good about adding extra little notes, right? And indicating where you thought maybe you had done something wrong. So science, we always justify, right? And draw conclusions.